Okay, thank you very much. Great pleasure. Thank you very much for inviting me. It is really nice to follow development of neuroscience here. For a long time, neuroscience in Wrocław was more like a desert, and suddenly, surprisingly, in an entirely new place, neuroscience is flourishing. Considering that this is Łukasiewicz network, which means more on the applied side, I decided to talk about something which is not really our main research interest. It's kind of a spin-off of our main research interest. Okay? I've always been driven by just the curiosity. Okay? But if an opportunity comes to apply some results of this curiosity, or actually doing something applied, translational, by itself may satisfy curiosity, I guess. I will show you how we got into this, where we are now. And we are now, at the very end, is collaboration with a company that went through phase 1 clinical trials and now is preparing phase 2 clinical trials. Everything started, Tomek is right. So my story is that, yes, indeed, I was doing PhD in experimental hematology at the military hospital. I worked at the children's hospital in pathology department in Warsaw. Then I was a postdoc in the US. But PhD as a so-called extern, I defended at the Nencki. But for the story and for my scientific development, which is one of the issues I understand also for those talks, is scientific career and development. Also, this postdoctoral stay, though in an entirely different field, was very influential. It was namely in experimental oncology, cell biology, cell proliferation. The topic we worked on was how the cells that are so-called quiescent, non-growing G0 phase of the cell cycle enter the cell cycle. And they go through G1, GAP1, and then there is S phase, DNA synthesis, GAP2, G2, and mitosis, cell division. And it happened, my mentor was Renato Baserga, and this was one of the first 3 labs in the world that started to look, could it be that this transition from quiescence to the cell cycle, as measured entering S phase, can be driven by patterns in gene expression? We are talking 1984. Okay? So I was involved in those studies, and indeed, what happened, that first of all, the genes were identified that drive G0S transition, and that were actually first shown to be activated, and then for some, like I showed for c-Myc, they are critical. I was doing so-called microinjection of proteins or RNA or antibodies. I could show indeed that certain proteins are functionally involved in the process. But even at that time, first half of 80s, at the time when I was just about to graduate, as Tomek said, 81, I got fascinated mostly by the issue of mind, how the mind is embedded in the brain, how the brain produces mind. But I thought, okay, it's good to have a kind of diverse background, got all these degrees that are necessary to be independent, and then I could follow whatever I want. And this really happened. So when 86, when I came back from the US and started to work at the Nencki, I came to the idea, can we apply molecular biology to this issue of brain-mind relationship? Using learning and memory as something that we can study in experimental animals, that is very fundamental attribute of mind. One of some, but very important. And at that time, molecular biology is not like that. It was more stricter defined in a more narrow sense as a structure and function of a gene. So talking about molecular biology was the question, okay, what are the genes? What do the genes have to do with this learning and memory. So again, following what I was doing on the cell cycle. So let's do the first simple experiment. Could it be that during some mind operation, some function of the brain, could be change in a gene expression? At the time was unheard of. Nobody in the fields of neuroscience would consider changes in gene expression in response to changes in the environment. Absolutely unorthodox. So the very first experiments we've done with two of my friends, now retired professors, Wojtek Danysz and Janusz Siedlecki, was we took the rat and injected just beneath the hippocampal formation into the brain glutamate. Glutamate was at that time being recognized as a neurotransmitter, was still not obvious, but for sure was also shown to be important for learning and memory, because antagonists of glutamate somewhat prevented learning and memory. So we did this experiment, and this is what is shown here. This is so-called Northern blot that measures RNA level. There was no PCR yet at that time. And you can see that this is the level of c-Fos mRNA 15 minutes after injection of glutamate just beneath the hippocampus. It's a massive and transient, is gone by 6 hours expression. This is for control of loading of this. More or less, at the same time, second half of 80s, this gene c-Fos, the protein encoded by the gene, had been identified, recognized, and understood as being a transcriptional regulator. And in essence, what we know now is that c-Fos dimerizes with a protein mostly from Jun family, forming so-called AP1, activatory protein transcription factor, that recognizes this sequence, specific sequence in DNA. Yes, Michal. So cut the very long story short, we follow two paths. One was unbiased approach. We use all the possible ways people were using at that time to identify differential library, RNA display, and so on. And the other was educated guess approach. Okay? I had this idea that why, being biologists, why not consider that any phenotypic change may relate to similar biochemical processes at the very beginning. I call them cell activation. Even published a paper called Molecular Biology of Cell Activation. And c-Fos, c-Myc were those players I was looking for. And c-Fos was a bingo. Because I did previously work on c-Fos in the context of cell biology and so on. So this was one. So anyway, so this dimer binds to DNA, regulates gene expression. Okay, so what was important at this discovery is that glutamate is not only acting on receptors or the synapse, whatever, but actually it leads the signal all the way to the cell nucleus. And the very fact that this is transcriptional regulator open a whole new world, because you would say it's a conductor in the orchestra. You may now consider that the gene expression programs are being regulated, not a single gene, because it's a transcriptional regulator. This injection of glutamate is cumbersome, it's not nice. You have to drill a hole into the skull, put the cannula, and so on. So it happens that we started to collaborate with two friends at the Institute of Pharmacology in Academy of Sciences in Krakow, Władek Lasoń and Rysiek Przewłocki, and they exposed us to a model of kainate. Kainate is an analog of glutamate. It activates so-called ionotropic AMPA and kainate receptors. But the drug can be just injected EIP intraperitonially. In rats, 10 milligram per kilogram. In mice, more. We started with rats. And then the kainate activates the ionotropic AMPA kainate receptors that depolarize neurons, that releases glutamate, that activates neurons, that release glutamate, that activate neurons, and so on. It's a lot of activity, and in result, is a status epilepticus, massive burst of seizures, and then development of epilepsy, which is important. So first thing is the excessive neuron activation. This is shown in the hippocampus. Okay. Then is so much activation that the CA1 corneal ammonis and CA3 subfields of the hippocampus die, partially through apoptosis, which we also incidentally discovered in the 90s. But the dentate gyrus survives. And the dentate gyrus gets projection, so-called perforant path, from the entorhinal cortex. Also dies out. Sends projection, so-called mossy fibers, to CA3. The target is gone, and what they are getting the information from, perforant path, is gone. Okay? So these poor guys at the dentate gyrus, granule neurons so-called, they do some aberrant plasticity synaptogenesis on themselves, because they have to do something with these axons and dendrites and so on. And this is probably the very basis of this seizure focus. So this is called aberrant plasticity, changes that support this seizure. And then there is also glial reactivation. Okay, in this model, we looked, okay, c-Fos is there. Okay, we also confirmed that after kinase, c-Fos is there. It's a bit later, the maximum. It's not 15 minutes, it's 6 hours, because those are these recurrent seizures, recurrent activation of neurons. But this was a question, okay, what about the functional form? A functional form is AP1. AP1 could be measured. At that time, we worked a lot with radioactivity, radioactive probes. In this case, we took a piece of DNA, 22 base pairs, yes, 22 mer. We had labeled with radioactive phosphorus, but in the middle was this sequence recognized by AP1. And then we had the protein extracts mixed with this, run through the gel and so on. And it's called electrophoretic mobility shift of psi or gel shift, in which we see additional band, we see that reflects the binding of the AP1. And you can see that there is no much increase after saline injection, but there is increase in 2 and 6 hours after kinase injection. There is decrease, there is 72 hours increase, and this was actually led us to the discovery of apoptosis. So it's an active process at 3 days. And then this technique can be further refined. This complex that is here, which is this oligonucleotide bound to protein, yes, can be reincubated also with antibodies against specific proteins we may expect to be in this complex. And this additional shift called super shift. And then it shows that c-Fos was included in this complex and some other components. Actually, there's not only c-Fos, there is FOS-B, FRA1, phosphorylated antigen, FRA2, c-Jun, Jun-B, Jun-D, all those guys are there. We are focused on the c-Fos. Okay, so now we came to the questions, okay, we have this, the glutamate receptors activation results in a gene expression. The gene codes for transcription regulator. Transcription factor is also increased. Okay, next step, what is being regulated by the AP1 under these conditions? Was, again, curiosity, next step. And then we tried different attempts and failed. Fortunately, a group of USC3 in Weizmann Institute were more successful with Ellen Nedivy as the first author. And then identified and published 93 something like 51 genes, some of them known, some of them unknown, that could be activated in the dentate gyrus of the hippocampus after kinase injection. Bingo, exactly what we wanted. So then to answer Michal's question, then again was an educated guess. So I asked Yoav, send me several, we got 5 gene probes, because I suspect them that they could be, maybe, AP1 targets. Okay? And the most successful, we were with TIMP1, which is an acronym for tissue inhibitor of matrix metalloproteinases. This is quite a busy slide. A study we done with Jacek Jaworski, late Irek Biederman, really showing that indeed c-Fos AP1 regulates TIMP1 gene in activated neurons. First of all, so we analyze whether in the gene regulatory region is this AP1 binding sequence, the one I was showing in some of the very first slides. Again, interestingly, we are aided in this case by a colleague from Krakow, Tomek Kordula, from Alexander Coy group, who cloned rat TIMP1 gene. Okay, we also got transgenic mice that had a PIS regulatory region of TIMP1 gene fused to beta-galactosidase as a marker. Okay, then we also first actually we looked whether TIMP1 is activated to confirm whatever C3 and colleagues did. And indeed, you see 6 and 24 hours, there's activation of c-Fos mRNA after kainate. But then, if it is regulated by c-Fos, it should follow, and the mRNA of TIMP1 should be where the protein of c-Fos is, yes? This was kind of reason. So what we did, we use another model of seizure-evoking agent, which is pentylenetetrazole. It operates different way. It doesn't work on glutamate receptors. In contrast, it blocks GABA A inhibitory channel. If you block inhibition, the excitation takes over. There's an imbalance. But in this case, it's one massive burst of seizures. It's not like kainate, this recurrent seizures, repeated seizures. No, one seizure, actually. So this was easier to see the sequence of events. And you can see here that TIMP1 indeed follows the c-Fos. We also looked here with kainate whether this TIMP1 mRNA expression is dependent on the previous protein synthesis. c-Fos has to be produced from mRNA and protein. So we use inhibitor of translation, cyclohexamide, and we abolish this. Okay. Then we looked whether this, I told you this interesting very question is, they should correlate in a pattern of expression with c-Fos protein TIMP1 mRNA, but in situ hybridization and immunostaining, we could indeed show this kind of colocalization. Very primitive ways at that time. In mice, it does happen in mice. Indeed, in mice, when we treat them with kainate, we also see this accumulation of beta-galactosidase that is driven by the regulatory region of TIMP1, but again, selectively in the dendrogyrus. Very good. And then we also did experiment. This was at the time that just coming back to Tomek and his master thesis that he did in my lab, just to culture neurons was a big thing. Okay, so I had a PhD student, Isa Figiel, who developed the first and the only culture system for the dendrogyrus granule neurons. And using this kind of culture, because this was, as you see in the granule neurons, we looked whether mutation in the, so whether this construct where we have the fluorescent-driven, GFP-driven, the TIMP1 promoter, whether it's activated by glutamate, and it is activated by glutamate, and then whether this can be abolished if we mutate ApoA, it's abolished. So there's a number of different, there were some other pieces of experiment, gel sheets and so on. Anyway, it shows that TIMP1 is a target. Okay, satisfying curiosity. But tissue inhibitor of matrix metalloproteinases. What the hell? What are matrix metalloproteinases? We never heard of before. Okay, so the next step was to look for the matrix metalloproteinases. And it turns out that TIMP1 is especially eager to interact with one of those MMPs called MMP9. There's over 20 of them, and they have these numbers. And it operates like this, that when the MMP9 is translated, produced, packed into vesicles, it is then it's not much better. It is packed into vesicles and then is released. It operates extracellularly mostly and is released in non-active form with a propeptide, 10 kilodalton, It's about 100 kilodalton molecule, 10 kilodalton covering enzymatic active site. It's released in a latent non-active form. And there are some other guys, like plasmin system proteases, that cut off the propeptide, and then the activity is unleashed and cuts through something that is around. But you can imagine releasing protease that is actually not very specific to that protease. It's quite, it cuts through very different proteins in the test tube, might be very detrimental. Okay, you don't want to destroy everything there in the extracellular matrix surrounding the cells. So then the TIMP1 comes within minutes, we show within a few minutes, is actually blocking the MMP9 activity. So TIMP1 blocks MMP9. Okay, so we go further. MMP9. Could MMP9 have something to do, with this kainate thing? And indeed it does. So it's great work of Leszek Kaczmarek. It actually opened this field of MMP9 plasticity, to cut the long story short. So what was done, the MMPs, among all these 20-something MMPs, there are two guys that can cleave gelatin. Gelatin is the natured collagen. It's a protein target. Those two guys that have numbers MMP2, MMP9 are called also gelatinases. And it's a very specific reaction for them. Now, it is possible to detect gelatinase activity by two ways. Either run through the gel, protein gel, full of gelatin, like the Lemley standard SDS, beta-mercaptan, or whatever, and then remove all this nasty stuff, and the enzymatic activity recovers, cuts through this gelatin, where is it? Then the gel is stained with Kumasi, everything is dark blue, except translucent buds. So it's called gel zymography. The other is in situ zymography. There's a reagent called DQ, dye-quench gelatin, that is a gelatin that is overloaded with fluorescent groups in such a way that they quench each other, so does not emit fluorescence even if properly illuminated. But if there's activity, the fluorescent groups separate and then can be visualized under the microscope. So this is the in situ zymography. After kainate, you see a massive increase of a signal around the dentate gyrus, cell bodies, and even dendritic tree. Then Arek was painstakingly collecting material from slices, ran through the gel zymography, and he could show that it's actually MMP9 because it was of proper molecular size, and there were these two bands. One is the Pro form with the propeptide, another the active when the propeptide is cut off. Then Grzegorz Wilczyński, by immunogold staining at the electron microscopy level with the antibody, could show that MMP9, these are the gold particles, 10 nanometers gold particles, visualized in the electron microscopy, could be present at the postsynaptic domain of excitatory synapses. Why we know that there are excitatory synapses? Because they are asymmetric. Symmetric synapses are GABA synapses. But the excitatory synapses, they have a lot of this PSD stuff here, postsynaptic density. And this is presynaptic, these are vesicles of glutamate, and then this is the postsynapse, and the MMP9 is there. Moreover, Grzegorz developed a very fantastic in situ zymography, high resolution method, in which he could show that this is in blue is a dendrite stained with MAP2. Then in red, there are so-called dendritic spines that harbor excitatory synapses that extend from the dendrite. They are stained with NMDA NR1 subunit. And then on the top of them, you see the green the in situ zymography signal. Moreover, also Filip Konopacki, for the young people, each of those is like the PhD. Each of this figure is one PhD, more or less. This is Maciek Gawlak, PhD. This is Filip Konopacki. Arek was doing PG on something else. Anyway, Philip showed this is that this signal of this increase of the dentate gyrus localized signal of MMP9 concerns also MMP9 mRNA. And in response to kainate, MMP9 mRNA is being produced and then goes into the dendrites. This is the staining of MAP2 in green, and the red are dots of MMP9 mRNA, and these are cell bodies. This actually led to, I'm not going to talk about, we also showed that MMP9 is locally translated. It goes to the synapses and is locally produced at specific synapses in response to glutamate. And then we also identified some substrates, these proteins that are at the synapse, and one of them was the beta-dystroglycan was shown to accumulate the cleaved product after the kainate. Okay, so now we go to the epilepsy. Because I told you that the status epilepticus, the kainate may provoke status epilepticus, and status epilepticus may develop epilepsy. Epilepsy is a major neurological disorder. 50 million people suffer from epilepsy. You have these definitions here. The problem is that something like 2/3 of patients with epilepsy, they may have the disease controlled by anti-epileptic drugs that prevent and enhance neuronal activity. But about 1/3 is not cured. Another way to cure epilepsy is the surgery. Identify where it's located in the brain and just cut it away. Especially by Penfield at MNI, Montreal Neurological Institute, champion for a long time. But then there is also a process of development of epilepsy, okay, called epileptogenesis. It's both, there's something that initiates epileptogenesis, and then epilepsy may aggravate itself through epileptogenesis. And one of those main reasons, main conditions, pathological conditions that provoke epilepsy through epileptogenesis are traumatic brain injury and stroke. So we have again this problem that epilepsy may come from the defined reason, traumatic brain injury or stroke, but also they may have infection mechanism, may have immune things, genetic and so on, like tuberous sclerosis, a very known case provoking epilepsy. And also Fragile X mental retardation syndrome is associated often with epilepsy. But I want to focus on the TBI and stroke because those are the major causes of epilepsy in adults, and especially in well-developed world, because you see 47% are false, just. They might be, this is 14% are traffic incidents, might be assault, may happen during the war. In general, traumatic brain injury is something that, I will show you the animal model, something that does not really may destroy directly the brain, but touches on a brain surface and then produces cell death. And stroke, you know. And now you see in this figure that especially this is the status epilepticus. If someone undergoes a status epilepticus because of drinking alcohol or something, it's very rare, but it does happen. Almost 50%, over 40% of such people develop epilepsy. In the case of TBI and stroke, 5 to 15% of people develop epilepsy over like 1 year or 2 years. Depends where it's located is the damage and how big is the damage of the brain afterwards. Okay, so having shown that MMP9 seems to be associated with epilepsy-evoking conditions, kainate status epilepticus, okay, why not test the relationship of MMP9? So what we did the functional experiment. We used transgenic animals, either missing MMP9 function because of the gene knockout, mice, we got them from Zena Webb from Stanford, or we produced in the lab, thanks also to effort of Witek, but this was PhD of Marcin the gene was under the control of synapsin-1 promoter, and this was active form of a protein encoded by this gene. So it was selectively only in neurons and was having much more of this MMP9. And what happened was that here we use, now we are much more clever, I'll show you, but at that time we have not been so clever. We use a model of so-called PTZ kindling. I showed this pentylene tetrazole that blocks GABA A. I showed you that you put this 50 milligram per kilogram and the animal go into seizures, mice and rat. But if you give like 30, 35 milligram per kilogram, not necessarily there are seizures, okay? But you will repeat this injection every 2 to 3 days, and eventually they cease after this low dose. So every dose changes something, produces some plasticity, okay? And this process of developing of this change is called kindling. It's electrical kindling, and this is called chemical kindling. So we use this model, and what we show that if MMP9 is not functional, they are sluggish and less responsive in this kindling model. And then in contrast, where you have excessive MMP9 function, they are more prone to develop epilepsy. And then also show that in these knockouts, one of the hallmark of epileptogenesis, so-called sprouting of the mossy fibers, is also diminished. So we provided genetic proof of concept that MMP9 contributes to develop to epilepsy in this model. More recently, we turned to more clinically relevant conditions, which is TBI. And in animals, there's a machine, it costs 10,000 euro, that you open the skull, and then you kind of, how to say, you touch the surface of the brain, you put something, there's a special, I don't know how in English, special device that knocks on the surface of the brain, more or less. And this imitates TBI. It's called controlled cortical impact model, CCI. And what happens is that within days up to weeks, the underlying cortex just dies out after this procedure. There are some changes in the control surgery. There's some indentation of the cortex, but there's not long-lasting change. So using this model, we first looked whether MMP9 is activated. This is gel zymography, this blue, and this is translucent band. And we looked at the ipsilateral cortex, so around the injury site, ipsilateral hippocampus, just beneath this dying cortex, but hippocampus is more or less okay, and contralateral on the other side of the brain. And what happens is, these are the numbers here and graphs, that there's a massive activation, especially in the ipsilateral cortex, around the injury site, of MMP9, okay? And also, in the hippocampus, there is also activation of MMP9. And it's much less, if at all, on the contralateral side, very little, if at all. And the sham surgery produces also some effect, but much, much weaker. Okay, so TBI provokes massive transient, this will be very important. We are talking up to few days, hours, through 1 to 2 days, increase in MMP9 activity. And then we looked whether in this case we have a contribution by genetic means of this MMP9. In this case, we actually used knockout mice, those from Zenaware, but also we got from colleagues from Greece, Atinia and Effi Libari from Athens. They produced transgenic mice with overexpression of MMP9. So everything is on mice now. And we first looked development of epilepsy. And the model is not perfect. It's like 10% of mice develop epilepsy after the CCI. Like humans, I told you, few to like 15%. So this is not very something. Okay, doesn't like this. So it's from 10% to 6% decrease in the knockout. But if you use overexpression, it goes, this is different white type, so these are different numbers because different mouse traits, from 16 to 62%. It's a massive, more MMP9, more eager to get epilepsy. This is the number of epileptic mice, this is number of seizures per week, more or less the same result. Then we also use so-called PTZ threshold test, another measure will be important later on, which is not as great, but still okay. It means that we provide them with a lower dose of PTZ, of pentylonetrazole, and we see how fast they can go into seizure. If they go fast into seizure, they are more prone to seize, okay? So this is also. And this is more or less the same thing, that missing MMP9 reduces chance or the severity of seizure, increase MMP9 by transgenic means, enhances this thing. Okay, and then, so we had genetic proof that MMP9 plays a role in epileptogenesis. There is no drug in the world to target epileptogenesis. Okay? There is no such drug. So why we don't try? I told you at the beginning that I started with this oncology field. I a bit followed this. And this was very famous. MMP9 was very famous, other MMPs as well, in the tumor studies. In fact, something like tens of billions, dollars, euro, whatever, pounds, were lost on the clinical trials to study MMP, including MMP9 inhibitors. Because MMP is, I told you, protease that is released outside the cells. So it's important for development where the cells has to go somewhere or release something, but it's also important for the cancer metastasis, when the cell has to go from one place to another place through different body membranes, whatever. It uses MMPs to dissolve things, okay? Like machete in the jungle. So people notice this in the culture, notice in animals, that when you put the inhibitors of MMP, like non-selective, but also for MMP9, you actually block metastasis. Great. Go to clinical trials. So they went to clinical trials. And after prolonged treatment with these MMP inhibitors, people suffered a lot of pain, musculoskeletal pain. Everything was abandoned. Left a very bad name for MMP inhibitors in the big pharma after losing these billions. Okay, but I had this idea, okay, with the epilepsy, the MMP9 increase also is only acutely. You don't need to block it chronically. You want to block the trigger for development of epilepsy. And the trigger is within minutes to hours after stroke or TBI. Okay? So I propose that maybe we can use those drugs, give them a new life in this kind of conditions, when you don't need chronic treatment. So this is what we did. And we decided, this was still with a company called OncoR&D. Vice president was Dr. Staszek Pikul, who spent years on MMP inhibitors in the US, working in the pharmaceutical company. I was very unhappy about this. Didn't want to do this work, but somehow I convinced them and his boss, Marcin Szumowski, the CEO of OncoR&D. And we got a grant. And we looked for some inhibitors of MMP activity that were used for cancer. But the first question was, could they penetrate blood-brain barrier? Could they operate in the brain? Okay. For this, we got Ania Konopka to work with us, and she was looking with the mass spec into brain extracts, whether they can be there. And one of those, called marimastat, indeed was found in the hippocampus and cortex after this injection. Then we wanted to know, okay, the guy is there, could it act on MMP9? And now it's not an easy question, how to see how we can affect activity of the enzyme. And what we invented as a way, I think the only possible, is see whether it prevents cleavage of its target. Okay, because we cannot put on a gel because they will dissociate through electrophoresis and so on. So we use another target. Emilka Rejmak, this was her PhD actually again, discovered that the Nectin-3 is a target of MMP9. If you have neurons in culture here, you treat them with NMDA that activates glutamate receptors, special kind. You have a massive release of a fragment of full-length Nectin-3, so-called small fragment, that is entirely blocked by MMP inhibitors. And here it shows that just treating these extracts or cells with active MMP9 also produces some cleavage products. So then we looked whether in the brain, okay, this marimastat, the MMP inhibitor, broad spectrum, but also affecting MMP9, can somehow prevent this cleavage, and indeed it does. Okay, so marimastat is not abolishing this, but is significantly decreasing the cleavage. And then we looked whether this marimastat affects seizures, development of epilepsy. This was very nice, complicated experiment. So there were mice, and they had implanted electrodes. We really recorded seizures. This is how it looks on the traces. We can see what is there. When we measured, or Basia Piet, who did this work, measured various things related, seizure occurrence, number of seizures, and so on, at different times, 2 hours, 24 hours, and 7 days after the injection of this. This was actually status epilepticus still here, because it was easier to follow than the other model. And what happens is just you can appreciate that there are some measures, seizure measures, after 24 hours or even 7 days that are decreased in these animals that were treated with marimastat. Okay? So this was very promising. We submitted a patent on this. Actually, we got this is still in a process. We just got recently Chinese patent on marimastat to prevent development of epilepsy. The others are still in progress, the patent applications. Okay, and then Staszek Pikol used to be vice president of Onco R&D, left this company. And more or less at the same time, I convinced him finally that maybe there is a hope for MMP inhibitor. It took me like 2 years to persuade him this difference between chronic treatment and acute treatment. And finally, no, light flashed. He got the idea. It took years, really. Okay, so let's try. And he started a company called Picralida. And the major project for Picralida was to follow our work with marimastat. The patent for marimastat expired and so on, so they call it PKL-021. They have their own name, Picralida 21. But it's marimastat, it's the same as was done. And we applied jointly for a big grant, over 20 million zlotys. We got a small part, but they formulated the drug to give to humans and so on and so on. It was called EpiFix, and this was a poster that kind of promoted this proposal. For that, our part was minor, and actually it was quite shocking that I worked on MMP9 since 2002, or actually worked for longer. We showed that it contributes by using genetic means to development of epilepsy in various conditions. This PTZ kindling, kinates evoke status epilepticus, TBI. For me, it was obvious that stroke is the same. But they found nothing in the literature on stroke. And then there was another grant we had, a European project with some colleagues. They invited us for another reasons, but they work on a stroke model that was refined by Nick Plesnilla in Munich, this Stroke and Dementia Institute. I don't know if you heard the story. This is what Solo Jacques contributed 100 million euro to create an institute in Munich. So they work in this institute, and they develop a special procedure to keep the mice alive. Because after stroke, definitely most of the mice die within a few days. Especially the model which we're using, the major model, which is major carotid occlusion model. So nobody really could look for epilepsy to develop later. So we had the model to keep them alive. So we looked here, and again, this is this PTZ threshold stuff. On the seizures with implanted electrodes, the picture is not so great, actually. But here again, there is the knockout, and there is a transgene, and the knockout somewhat prevents developed susceptibility to seizures, where the overexpression produces. And this is here, this is overexpression, and also the drug also did more or less similar work, not maybe as good as the knockout, but still produce some effect. So it seems that indeed we have something to work on. Okay, so they went further, as I told you at the very beginning. Last year, at the end of this year, they completed the clinical trials phase 1, looking whether the formulation, this is a major thing, I didn't realize, it took millions to formulate the drug that to be given to patients in a safe way. Anyway, they managed, and they did the study. The drug is well tolerated, it does not produce any adverse effect, given with 3 doses, like 50 milligram or even 200 milligram per kilogram. In humans, when they were studying this drug, they found that 900 milligram per kilogram, single dose, was absolutely safe and perfect. The problem was with repeated doses with the chronic treatment. In mice, we are doing like 9 milligram per kilogram 3 times, and it was still effective. So we still are thinking about the dose, because I'm saying maybe 50 milligram will produce even better effect that you can see here. In the meantime, I generated a competition to us. That's a kind of digression. It means that I got in contact with a small company, a spin-off from the Institute of Biomedical Research in Barcelona. The company was called iProteos, now merged with others, is a different name. And with 2 people there, Teresa Tarago and Roger Prades, I kind of inspired them. Okay, they come from chemistry department at that institute, and they wanted to do some peptide-like agents for something. Said, okay, why you don't look for inhibitor of MMP9? So they spent years, and they tried to find inhibitor of MMP9, and indeed they found something that was effective. The first paper was collaboration with us, the nectin-3 cleavage was somewhat decreased with increasing doses of this inhibitor, which is shown here. And then now, without us, they progressed further and published a paper which is essentially with the same results as our paper with Bashapir, that they use this design inhibitor to prevent epileptogenesis in mice, and also got more or less similar results, that some measures of seizures were decreased after treatment of the drug. How far they progressed to clinical applications, I don't know. We are in a better position in that sense that we follow the strategy of drug repurposing. So we have more information about the drug. So the drug originally, Marimastat, was produced by British Biotech, and after this collapse of this clinical trials, British Biotech collapsed, and they did not release any information. Picralida had to repeat a lot of stuff that probably they have on file somewhere, but they will not reveal. Anyway, the MMP9 seems to be effective in preventing epileptogenesis, though I have to say we don't know at which level and where it operates. With Daniel Homak, a PhD student, he created this nice picture that will be a lot of different biological, physiological, and pathological phenomena to which MMP9 may contribute. It contributes to blood-brain breakdown that occurs after stroke or TBI. It contributes to reorganization of extracellular matrix. It contributes to synapse remodeling, which is our main interest. But axonal damage sprouting, in result neurodegeneration, but even neurogenesis. So we don't know which of those are affected. What are the mechanisms? Maybe all of them, maybe some of them. And also another warning. We wrote this review also with Kasia Łukasiuk some time ago. She worked with Asla Pitkänen, who is one of the worldwide leaders on epileptogenesis. She's in the University of Kuopio in Finland. That actually there's a number of proteases that have been implicated in development of epilepsy. We usually kind of overlook how much life is outside the cells, how active is this life of our outside the cells. So this is MMP9 here and TIMP1 here, yes? And this is pro-MMP9. And there are some other proteases, and even receptors could be, Asla Pitkänen was working on this PAR1 receptor and so on in the context of epileptogenesis. So anyway, the take-home message is that targeting MMP9 offers a viable therapeutic option to impair epileptogenesis. There is nothing else. I don't know if we are the most advanced in the world in that sense to find a drug. I think that the other people try. It's important for. But something really interesting, what way to pursue. So finally, I would like to thank. This was, as you've seen, this was long time effort through kind of even decades. There were many, many people involved. We got a lot of different grants, especially European grants, to work on those similar area related to MMP9. Even if still my main interest is MMP9 in the mind and synaptic plasticity. Okay, this was our main scientific journey. And then there are people in the lab, and this is Basia Piet, who contributed a lot to this TBI. Daniel Chomiak, I showed his picture. Agnieszka Koszczewska to this stroke research recently. Emilka Rejmak I mentioned, and Piotrek Michaluk, who did his PhD on MMP9 with me, not on these models, but after 7 years at the UCL, got back to Nencki and still collaborates with us. Okay, thank you very much for your attention.
Thank you for this wonderful lecture. It was a great pleasure to listen to it. Time for question, comments. I start with Michał Malewicz. Michał Ślęzak will be next.
Thank you, Leszek. Wonderful story. I particularly like the fact that we have now a drug candidate for which we know how it works, which a lot of times is a mystery and obviously doesn't help with further development. But I wonder whether moving forward, there are going to be challenges with regards to definition of the actual patient group which it helped most, right? It's like setting up even the phase 2 clinical trial already, at least from sort of my naive view, would be a little bit challenging because you don't have a classical disease, and sort of indication definition would already be a little bit challenging, right?
Okay. So the idea is one, the simplest. Okay, everyone who undergoes either stroke or TBI gets to the hospital, and within hours after the insult, gets the drug. Shouldn't really harm. Then we have to wait some years to see how many develop epilepsy. That's the simplest. I believe that we can identify more susceptible group. You are absolutely right. Remember our results that the transgenic mice with MMP9 overexpression, they are much more susceptible to develop epilepsy after TBI and also after the stroke. And I would rather, maybe in the clinical phase 2, we will single out this population and see what is the efficacy of the drug toward them. And there are at least 2 ways to how to single out them. One, we showed, like for schizophrenia also, and was also showed for some form of autism, that there is a correlation of MMP9 in the blood plasma and some brain function. And There are increases of MMP9 blood plasma that may stem from the inflammatory situation, even in viral or bacterial infections. Okay, so then there is increase. So it would be nice to see, just to measure in the blood plasma in all those people, the MMP9 and see whether those with higher levels may be more susceptible to develop epilepsy. One. And this will be done, I understand. That's simple again. A bit more complicated, but also I think should be done, is to use natural experiments that have been done on humans with MMP9. MMP9 has recognized gene polymorphisms. Interestingly, functional gene polymorphisms that do not affect the structure and function of the protein, but they do affect the protein level. Especially very famous is minus 1562, 1562 base pairs in front of the transcription initiation site. In this polymorphism, about 25% of us have a polymorphism that results in the higher production of MMP9, and 75% with lower. So again, I would expect that if someone falls into this polymorphism with higher production, there's much greater chance to develop epilepsy, to narrow down this group. But it's not really harmful. By the way, there's another interesting thing about this MMP9, what they discovered, that there's a very selective group of patients for whom it's worth it to give marimostat. It's with people who were, who got snake bites. Over 70% of snakes use MMPs to produce the damage. And apparently it's nothing better. So at least this 100,000 people worldwide could have this. Okay.
Thank you, Michał.
Hi, Leszek. Thank you for the really inspiring story. On one of your slides, you showed, one of your last slides, you showed that if you look on the knockout mouse and the mouse with overexpression, the basic level of the measure of the epilepsy is basically the same, which would mean that whatever MMP9 does needs to be triggered. So in other words, as I'm thinking about it right now, the substrate which is critical for you, for the epilepsy is not in the brain, but rather based also on the risk factors you showed, might be the result of breaking down blood-brain barrier. Is that correct thinking?
No, first of all, MMP9 is not there. MMP9 has to be triggered. Normally in the brain, you do not have MMP9. Almost zero. And there is this massive activation by various triggers.
Yeah, but in your overexpression model, right? And still there is no increase in baseline epilepsy measures, right? Which would mean that it still needs to be triggered. So I think it was 4 slides back.
I'm saying this.
Right? No.
Yeah, I think that this is much better in that sense. Yes? In which sense you mean?
I was talking about the neck. Well, here.
But here, okay. Here,
yes,
we do not have really epilepsy. We have susceptibility to seizures after triggering PTZ.
Exactly, but what I'm saying is overexpressing MMP9, only overexpressing MMP9 does not trigger epilepsy, right?
Overexpressing MMP9.
You have OE sham.
You
right?
mean this sham condition?
Yeah, OE sham is the same
sham,
level as
sham.
the knockout sham, right?
Yeah, just having MMP9, yes, does not. I can tell you because also our rats are not epileptic when you look at them, yes? They were not epileptic.
Right,
so, yes.
so there needs to be a trigger. So is it a substrate that needs to come to MMP9? Is it the right substrate which comes, for example, from the, I don't know, plasma after the breakdown of blood-brain barrier? Because it also correlates with what you said about inflammation, right?
But I don't know. Yeah, the thinking is interesting, but I don't know whether this MMP9 that is overexpressed is, for example, synaptic. If this is synaptic plasticity, you have to increase this MMP9 at the synapse. This MMP9 that is in the overexpression can be kind of a tonic. Secondly, I guess if there is increase of MMP9, because this was a great disappointment, the rats, because they have massive increase of MMP9 that is not further potentiated by kinase or so, and they virtually have no phenotype by themselves.
It is also overactivating form of MMP9, which is not necessarily translated. So the model, in the sham model, where you have the overexpression animal, you do not necessarily see how to activate the MMP9 protein, because what you have is only overexpression of mRNA.
No, but you see the protein. By gel zymography,
Only after induction.
now, by gel zymography, if you take the brains of those rats, you have much more of MP9, both forms, active and latent. So, and they have no phenotype. So again, this is biology. You would expect that such dangerous guy, like protease outside the cells, is somewhat blocked, should be compensated. And then the trigger releases this. Still, it has to be something, because they should die. If they would have overexpressing MMP9, they should die through development, because you damage extracellular milieu for those.
For example, if you were to inject, let's say, 3 plasticity, you have
No, that's again, that's so simple, because nectin-3 is in the membrane. It's a transmembrane protein. Okay? So, and there are other targets. Okay, we identified.
What I mean is that the cleavable targets of MMP9 may be the one. What I'm saying is that perhaps it's the cleavable targets of MMP9 and not so much the expression of it itself, but the activation of MMP9 in some ways that we don't understand.
Yeah, but I'm saying activation is seeing the active form. So we see in the transgenic overexpressing animals the active form. So the MMP9 is there active, but still not much happens. Okay? So yes, maybe it has to be substrate that appears there, or maybe you release MMP9 that is under those conditions somewhat blocked. Okay? Because the organism cannot accept to have enhanced MMP9. This I am absolutely sure. It's so important in various processes that if you would have even development of the brain, if just neuronal MMP9, it still will be damage to the brain. And I'm saying the overall, we expected much more of a phenotype, and we didn't see much. Only the triggered phenotype like epileptogenesis. But when you look at those rats, we did a lot. It was whole PhD of Marcin. There was practically no phenotype.
Okay, thank you. Maybe one more question, Mateusz Kucharczyk.
Thank you. Excellent talk. Thank you very much.
Thank you.
Great overview on MMP2 and c-Fos as well. I have a question. So you mentioned for the MMP2 inhibitors used in clinical trials for cancer, the patients were having a painful phenotype. Was that a musculoskeletal pain?
Yes.
Right. And do your MMP2 knockout animals also have painful phenotype?
I don't know.
All right. And one more question. Is this MMP2 model you have of the knockout inducible, or is it MMP2 knockout from the developmental
This is
stage?
MMP9.
So MMP2, yeah, MMP9,
this
sorry.
is the unconditional classic knockout.
Right, so from
Somehow
the development.
nobody, because it's important for so many things that maybe people were afraid to put all the efforts, but someone will kind of beat them. But yes, now we do have the conditional knockouts.
So I'm asking all of this because it reminds me very much of a story that I was from the very beginning involved in with tanezumab for NGF treatment. This is also a very typical situation with musculoskeletal pain in patients, and that's why the tanezumab at phase 3 clinical trials was axed for Pfizer. MMP2, as you know, and MMP9, both gelatinases are involved in the maturation of DNF growth factor. And I'm just thinking if there might be a link for the musculoskeletal pain that is developed for the inhibitors of MMP2, or perhaps knockouts as well. That would be very interesting to see in the knockout itself, and the link with the maturation of the NGF in the periphery. if you have a developmental model and compare it to the inducible one, there will be also a very important thing for the NGF. It's crucial for the development of nociceptors. So if you remove NGF at the early developmental stage, those animals will not only have no pain, they will be totally insensitive to pain, probably. Very similar to like congenital insensitivity to pain patients.
Yeah, could we? But we never work on pain, really. And
Could we get you animals from me?
and never worked on MMP2, so... yeah.
MMP9. No.
But I'm saying my main interest is MMP9 at the synapse, excitatory synapse, selectively.
I'm all in the periphery.
Yes. So we never work on the peripheral nervous system. By the way, it's even more complex because one of the targets we identified for MMP9 is pro-BDNF.
Yes.
At the synapse.
Yeah, that's the depression more like stories probably more
And
interesting.
plasticity in general.
Yeah.
Yeah, I agree. But
Excellent.
Claudio Coelho was doing this work on NGF and MMP9, yes.
Thank
I
you.
know Claudio.
Thank you. Sometimes during our discussion, we have some students and the young scientists who wish to ask questions, but they are a little bit afraid. So after a short break, you will have opportunity to chat to Professor Kaczmarek, who will be gladly answering your question. So let's thank Professor Kaczmarek once again for his wonderful lecture. Thank you.